Yeah, now it's good. Listen very carefully. So, see, first we have blood vessel, pathogen entered. Pathogen release some chemicals. Those chemicals are called as pathogen associated molecular substances. Okay. Now, a macrophage, a circulating macrophage got attracted towards the pathogen. Okay. This attraction is called as chemotaxis. First, chemotaxis. After chemotaxis, what happened? Now the macrophage will attach to the pathogen. It attached, attachment. After attachment, what happens next? Now engulfment happens. Engulfment of pathogen by the macrophage. After engulfment, the pathogen will digest. Digest the pathogen. After digestion, the macrophage will present the pathogen molecules on the surface. This is called as antigen presentation. This antigen presentation, whatever cell that can able to do phagocytosis and antigen presentation, such cells are called as phagocytic cells. Such cells are called as phagocytic cells. Some of the examples of phagocytic cells are monocytes, dendritic cells, then we have B cells. Even B cells can able to do this one. B cells. Okay. Few more are there, etc. So such cells are called as phagocytic or antigen presenting cells. So up to here is clear. So then see here we wrote already presentation of antigen. Antigen presenting cells present. This I need to connect. Now after this, listen very carefully. Every macrophage has, by the way, macrophage is a type of lymphocyte. Okay, so yeah, not directly lymphocyte, but okay, we can say okay, monocytes. Okay, so now listen. This macrophage has a cell receptor. That receptor is called as T cell receptor, TLC actually. What they wrote here. The helper T cell will recognize there is a receptor. T cell receptor. T cell receptor. So it has a receptor called T cell receptor. Okay. With the help of T cell receptor, now a helper T cell will read the antigen presented on the macrophage. This is helper T cell. After reading the pathogen, this helper T cell will be split into two types. What are those? Helper T cell 1 and helper T cell 2. This is what we learned yesterday. Right? Then helper T cell 1 will go for calling about cytotoxic T cell, whereas helper T cell 2 will call the B cell. B cell will request 1 week time. Within this 1 week time, it will try to figure out the type of antibody. Meanwhile, cytotoxic T cell will try to kill the pathogen. But cytotoxic, we know it's a cytotoxic in nature, so there is a regulatory T cell. Regulatory T cell. This is what we learned yesterday so far. Okay. Now listen very, very cautiously. The problem here was we understood there is an external pathogen and these things are recognizing. First of all, the immune system must have an identity system. How to differentiate or listen, I just want to tell you one thing. One terrorist came to the RIPANS, for example. Okay, a terrorist entered into the campus. Okay, now one security guard came in. Let us see. Now we kept a mandate in the campus that all the students must have their ID card. Okay, so what is the use of ID card? To identify whether you are a Ripanian or not, whether you belongs to our institute or not. That's why we given you the ID cards. Okay. Similarly, all faculties and the security guards has our own staff ID card. We have green ID card, you have orange ID card. This is clear, right? Now listen. So let us assume a scenario. We are going, one intruder came, one pathogen came, one terrorist came to the campus. Now a security guard catch the terrorist and he went to the department, to the administrative block. What we will do? Shall we going to kill the terrorist, or what we will do is, ideally, first, I don't know whether the security guard belongs to RIPANS or not, or he might be doing some tricks. I cannot even trust my own security guard if he don't have a ID card. First I need to read whether he is my authority or not, whether he belongs to my system or not. Only after trusting him, then I will trust whomsoever he brought. I cannot blindly trust anyone, right? I cannot trust anyone in this world. Got it? Similarly, in our immune system, we have two identity systems. Actually, there are three identity systems. I will write. Those systems are called as major histocompatibility system, MCHC, or this system is also called as human leukocyte HLC, human leukocyte complex system, HLA, human leukocyte antigen system. Okay, either HLA or MCHC, both are same. Okay, now this system has three classes. This system has three classes: class 1, class 2 and class 3. Class 1, class 2 and class 3. Is it clear? Yeah. Just a second. I need to cross check with my notes, then I will be back to you. Let me check here. Yes. I need one statement. Now listen. I got my statement. Listen. Now, MHC1 present in all cells of our body. This MHC cell receptors present in all cells of the body. Write this statement. This is important statement. Do you know what is HLA system, by the way? You know, right? Or you don't know? You might know slightly, but not much. Okay. All our cells have HLA1. All our cells has HLA1. Whereas only the antigen presenting cells, only AP cells has a different HLA. That is HLA2. Only antigen presenting cells has HLA2. Only antigen presenting cells has HLA2. Is it clear? Then HLA3 is dedicated to regulate something called complement system in the body. That I will discuss later. Complement system plays a very vital role, by the way. Complement system in the body. HLA3 will regulate complementary system. HLA1 is a molecule that present in all cells of the body, whereas HLA2 present only on the antigen presenting cell. Okay, that means our antigen presenting cell has HLA2. Am I correct? As per the statement, our antigen presenting cell has HLA2, whereas helper T cell will have HLA1. Okay, what I am going to do was, first I will read the admin ID card with my security ID card before checking his suspect. First we will read each other. Are you my staff? I am not going to mind about terrorist. First I need to cross check his identity. Are you my staff? At the same time, he too have the same right to check my ID card. Do you really my higher authority? If we both are not matching, first what we will do, we don't worry about terrorist. First I will kill my own self. I will kill my God, because he is a suspect here. Got it? This is the exact thing happens here. Now helper T cell first to read the macrophage MHC. MHC1 will read MHC2 before reading the pathogen. If they are not matching, they will auto attack each other. Got my point? Now listen. Now this HLA1, 2, 3, I forgot the exact one, DP, DQ, DR, ABC. I am not sure exactly which one is which. To my knowledge, HLA1 has a classification of A, B, C as a subclassification, whereas HLA2 has subclassification, that is DP, DQ, DR. So now what I was trying to say was, these are some sequences. of this arrangement of these HLA molecules, there are some sub-protein molecules. Now, each and every one of us has, just like our fingerprints, we have a different sequences of our HLA molecules. My HLA sequence is different from her HLA sequence. Even though I am O positive, she is O positive, but we have different immune fingerprint. I have a different immune fingerprint, she has a different fingerprint, they have a different fingerprints. We all have different types of immune fingerprints, self-identifying fingerprints. Okay, now listen. Sometimes in our body, sometimes some of our normal cells, say for example, this is a bone marrow, this is a bone and this is a hyaline cartilage and this is a synovial joint and this is one more bone. Okay, sometimes due to wear and tear, sometimes some of the cells in hyaline cartilage, they will lose their DNA, they will damage their DNA. Due to DNA damage, some of their MHC1 won't be expressed properly. The cell was damaged, their nucleus was damaged, so they can't prepare the protein, the self-identifying MHC1 protein. Now they lost their MHC1 protein. Now there is a nearby patrolling macrophages. You know, this symbol stands for macrophage in immunology textbooks. If you go to core immunology textbook, they will keep some symbols for various cells. Okay. Now see. Now this patrolling MHC, first it will see, it will try to read this cell. Now this cell won't show its ID card. Are you foreigner or are you self? Then the cell will try to say I am self. Then it will request the ID card. If cell not producing ID card, you know what happens? Macrophage will start digesting this. It will present our own cell as a foreign pathogen. Now helper T cells will recognize it as a foreign. Helper T cells will send a signal to the B cell. B cells will start producing antibodies to kill our own cartilage, leading to autoimmune disorder. Now you understood what is autoimmune disorders. See, our body attacking our own body because our cells lost their identity, self-identity, leading to autoimmune disorders. And this statement, whatever I said, this is rheumatoid arthritis, autoimmune rheumatoid arthritis. You know, right? Rheumatoid arthritis, where our own antibodies will attack our own synovial joints. This is the one. So now we understood the significance of HLA system. HLA plays a very vital role to identify self from non-self. They have their own particular way of understanding each other. They have their own system of understanding each other via HLA molecule or MCHC system. MCHC1 or class 1 present on all cells of the body, whereas MHC2 only present on the antigen presenting cells. This is what this textbook stated here. See, class 1 MHC1 molecules, it is found on every cell of the human body, especially responsible for presentation of endogenous antigens. Okay, you just remember endogenous. Whereas class 2 molecules, it is found only on B cells and macrophages and other antigen presenting cells. Only antigen presenting cells has class 2 and class 1 present on all other cells, other antigen, apart from APs, antigen presenting cells. Then this textbook discussed about antigen presentation. Antigen presenting cells have, the antigen presenting cell has class 2. Why it say class 2 now? Antigen presenting cells present a class 2 MHC molecule together with bound HLA and this activates. Sequence of events: helper T cell recognize antigen displaced on the surface of antigen presenting cell with the help of its own receptor called T cell receptor. Okay, helper T cell has a receptor. With the T cell receptor, it identified antigen presentation. Recognition of the antigen by helper T cell initiates a complex interactions between helper T cell and antigen. Okay. At the same time, macrophages will release. Yes, listen. This is important. Just a second. Now I'm getting confusion. MHC1 is found on every cell in human body, especially responsible for presentation of endogenous. MHC2 found on So do I miss the? Here we should have MHC2. This is one. Oh, I am correct. Oh, I was confusing. Sorry. Fine, fine. Now listen. One important step after this one. The important step was, okay, helper T cell read the foreign antigen. First they read their own MHC1 with MHC2. Done. Now, cells have their own way of communicating. I said, right? Now helper T cell will call the cytotoxic T cell. Is it a human to call? Does it have any walkie-talkie to call cytotoxic T cell? Then how it is communicating? We need to understand that. Okay? That is very important. There is a dedicated chapter for this particular cell communication. Any two cells will communicate each other by some signaling molecules, some chemicals. These chemicals are called as, can you guess what are those chemicals? Cytokines. Very good. Cytokines. Cytokines. Can you tell me what are the types of cytokines? There are 7 types of cytokines. This Shambhalingam given a very good cytokines table actually. I don't know whether I will get it or not, but it has a very good table. I liked it. I just want to show you that cytokines. Let me search here for the cytokines. Cytokines. Actually, I like this textbook. Yeah, here they give. See, types of cytokines. Write down very, very basic cytokines. There are 7 types of cytokines. There are 7 types of cytokines in the body. Cytokines are of 7 types. Oh, let me write this word. Somewhere I touched it here. See. Very good. I reach anyway. Types of cytokines. Understand it? Yeah. There are 7 types of cytokines. They are, just write the names. We need not to go there. Okay. They are interleukins. 7 types include 1. Interleukins. 2. 1. Interleukins. 2. Interferons. Interferons. 3. Tumor necrosis factor. Tumor necrosis factor. 4. 4. Chemokines. Chemokines. 5. Defensins. Defensins. 6. Yes. 6. Defensins. 6. Cathelicidins. 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 And 7. Platelet activating factor. 7. Platelet activating factors. So these are the 7 types of cytokines we have. Okay. Interleukins, then interferons, tumor necrosis factor, chemokines, defensins, cathelicidins, and platelet activating factor. Platelet activating factor. These are the 7 types of chemicals or cytokines. Clear? Now, I will let you know here. Somehow, the macrophage must call the helper T cell. It's not like a simple game, right? It might send some chemicals to call a helper T cell. It might send some cytokines. That is how the helper T cell came to know to read the pathogen via TLC. So, what cytokines released by the macrophage? We should know them. What cytokines released by the macrophage to communicate with the nearby helper T cell? That we will see here. See, they wrote here. Clearly they wrote. Helper T cell recognizes antigen displayed on the macrophage. Recognizing of antigen by helper T cell initiates some complicated reactions. At the same time, macrophages, the antigen presenting cell, will release interleukin 1, which facilitates the activation and proliferation of helper T cell. That means our macrophage released a chemical called interleukin 1. Write down. With the help of interleukin 1, the macrophage has communicated with the nearby helper T cell. Has communicated with the nearby helper T cell. Clear? After this, what happens? Now, the helper T cell will read each other. Class 1 will read with class 2 and they will check the pathogen antigen with the help of T cell receptor. Then this activated helper T cell will again release two types of chemicals to make helper T cell 1 and helper T cell 2. What are those chemicals? Simultaneously, here, role of helper T cell. So, role of helper T cell 1. Helper T cell 1 concerned with two substances, interleukin 2 which will activate other T cells and gamma interferon which will activate the phagocytic activity of the cytotoxic. Simple. This helper T cell 1 releasing interleukin 2 plus it releasing what is that? Gamma interferon to call the cytotoxic T cell. This is how the helper T cell communicated with the cytotoxic T cell via interleukin 2 and gamma interferon. These are the two cytokines responsible for communication between helper T cell 1 with cytotoxic T cell. Is it clear? Yeah. Next one, helper T cell 2. So helper T cell 2 concerned with humoral, that means antibody. It communicated via interleukin 4 and 5. Simple. Here interleukin 4 and 5 responsible for B cell communication, whereas 2 and gamma interferon responsible for the cytotoxic T cell communication. So now we are seeing the close connection between these cells. Okay. Why these things are important? Sometimes in some rare immune disorders in some patients, these cells will fail to release these cytokines. If cell fail to release these cytokines, we can't get, the immune system won't get activated. Or I will tell another interesting thing. Imagine I have taken CD4 cells in a test tube. I have taken CD4 cells in a test tube. Now, I was expecting my CD4 cell to convert to helper T cell 1 and helper T cell 2. How can I convert my helper CD4 cell to helper T cell 1 and 2? You know what I will do now? I will add a reagent to this test tube. What reagent I will add, I will add simply the chemicals released by the antigen presenting cells. That is interleukin 1. If I add interleukin 1 to my CD4 cell, this cell will get activated and it will be split into helper T cell 1 and 2. Okay? So, I am deciding the cell to replicate, to become what? Just by adding some chemicals. So now I am controlling my immune activity in the body by adding these cytokines. Now we are doing reverse engineering. Sometimes, for example, B cells, in order to activate the B cells, okay, we have taken a test tube. There are, we have through apheresis, cell separation, we have taken B cells in the test tube. Now my B cells want to become a plasma cell, activated B cell. What I will do? I can simply add these cytokines, interleukin 4. If I add interleukin 4, it will activate the helper T cell 2 to communicate with the B cell. So yes. So now see, there is some tight connection between cytokines and the antibody production. We thought simply B cells will produce antibodies themselves. Sometimes patient might having these issues, cytokines not working properly. We need to rule out. Now COVID. What COVID did was very interesting physiology. See, the COVID molecule, it's keep on doing something called cytokine storm. Cytokine storm, very, very interesting. So you have inhaled the virus, it's there in the lungs. Now what this, there is a spike protein, I think spike protein 1 or 2 of the COVID virus. This spike protein keep on activating the helper T cell to release gamma interferon. Keep on, especially the interleukin 2. This spike protein, activating the helper T cell 1 to release, this one, interleukin 2, keep on releasing. So what's happening? This interleukin 2 in turn activating the cytotoxic T cell. Cytotoxic T cell coming to the site, it's destroying the lungs. The patient getting more fluid accumulation in the lungs. Again this no more virus present in the body. Again, it's telling the helper T cell to release again IL-2. This becoming a frenzy now. Cytotox are even more attacking. No, you don't know what's going on. So since there is a signal, keep on destroying it, because that's what the immune system asking for. Actually, it's not immune system that is asking, but it is the pathogen that triggered the cytokine storm in the body. Now, if this is happening, if this going on, this vigorous vicious loop going on in the body, there is a one kill switch. The kill switch for immune system was steroids. You dump the steroids will damage all the cytokines. Steroids will remove cytokines from the system. That's why whenever patient having respiratory anaphylactic shock, when they are unable to breathe, when they have this sudden shock, immediately doctor will give some steroids. Steroids is the answer for every problem actually, but at the same time steroids will weaken the immune system. That is the mechanism of steroids. Steroids will suppress all the cytokines. They will remove cytokines from the equation. So immune system will be cool down. It will temporarily cool down. Got my point? So that is how the steroids works. And steroids themselves have another problems that we will discuss later. Yeah. So now we are understanding the deep mechanisms of cells and their interactions. The cytokines, the way they are communicating. These things, autoimmunity, and the self-identifying systems such as MHC1 and MHC2. Clear? Yeah. So I was not going as per the syllabus, because once you know the top concept, you can study yourself from any textbook. See, now all these things becoming clear to you. If you see cytotoxic T cell, you can simply understand the mechanism of cytotoxic T cell. This is activated by some helper T cell 1, then these things are happening. Okay, so you should know the concept, that's it. See, here there is a beautiful table. This table I was searching actually previous. Oh, this one you were. Why? Somewhere I touched, that's why it's not working. Anyway, now you know what are cytokines. There is one table actually. In the table you can understand all these things. Next one I want to discuss was one of the important topic. Tumor interferons. See, there is one topic like this chemokines, cytokines, tumor necrosis factor, interferons. Discuss about them and cell responses. The same thing, whatever I discussed in unit 3 we saw. Okay, then yeah. So if you go to the next page, there you have types of antigens that we'll discuss later. Immunoglobulins and their structures, types of immunoglobulins. What are the classes of immunoglobulins? Yesterday I given a simple mnemonic. What is that? It's a game of destruction. GAMED, right? IG, GAME and D, right? Those are the 5 classes that we'll see later. And monoclonal antibodies. Monoclonal antibodies. Do you know what is the concept of monoclonal antibodies? Any idea? You don't know? Listen, monoclonal antibodies. See, now after understanding the immune system, we are also exploiting the immune system for our own benefits. That's the beauty of understanding the concepts. Once we know how it's working, now we can tamper it and we can use for our own applications. Right? Like the way I said in this test tube example. I took just a B cell, now I need to make it into a plasma cell. I just need to add these cytokines and that will activate the cell and it will produce something that I was expected. Right? Yeah. Now let me tell you other thing. Undo. Is there undo in this? Okay. Okay, fine. Because this is a split screen, not the notebook. In notebook, that options might be there. Yeah. Yes. Where am I now? What I need to discuss? What I said before? Monoclonal antibody. Thank you. Monoclonal antibodies. So, what are monoclonal antibodies? Very, very interesting topic. Very interesting. Listen. We know about ABO grouping system. I hope you know what is ABO grouping. Tell me what is ABO grouping? Very simple. We forgot. Very good. Well done, well done. I appreciate you. Red blood cell, if it has a antigen, that is A group. If red blood cell has a B antigen, that is called B group. If red blood cell has both A and B antigen, that is called AB group. If red blood cell don't have these two antigens, that is called O group. Oh, thank God, now you know everything. Yeah. And if red blood cell has RH factor, that is a positive group. If you don't have RH factor, that is a negative group. Same. Positive, negative, positive, negative, positive, negative. So how many groups we have? Now tell me. 6. Oh my God. 8 groups. ABO, positive and ABO, negative. ABO, AB. Okay, 4, 4, 8. Simple. 8 groups we have. So how we identified this one? We did a very simple trick. I have taken a glass slide. Now what I did was, I have taken a known antibodies. I taken known antibody. First I have taken, and if you take a test tube, okay, and if you let the blood to clot, what you will get? The supernatant will be what after clotting? Serum. Yeah, serum. Okay, in this serum our antibodies are present. If you want to collect antibodies from the from the sample, you must let the blood clot, then you will get serum. That's why antibodies are also called as sera. Antibodies are also called as sera. Okay. And this therapy, antibody therapy, is also called as serotherapy. Do you know that COVID patients, recovered COVID patient blood, we will take his serum and we will give it as a drug, as a treatment to the COVID patient. Because these antibodies will protect the critically ill COVID patient. And this therapy is called as serotherapy. Why it is called serotherapy? Because we used serum. Antibodies extracted from the serum. Clear? So similarly, I am taking a known sera. What anti-sera I am taking? Anti-sera A. That means this will attach to these are antibodies that will bind to the antigen A. Then I have taken a drop of A antibodies. Simple. Then I have taken a known B antibodies. And that's it. Simple test. And I have taken a known RH antibodies or anti-seras. Okay. Now I will add a drop of unknown blood group. I don't know what is his group. I added 3 drops of blood. Now if I see reaction in this tube, what does it indicate? That means this person has antigens, and that is why this antibodies bind to the antigen and there is a agglutination. Right? And A antibodies didn't attach because this patient don't have A antigen at all. He might be B group. If he don't have RH, then this group will be B group. B negative group. If he has a reaction here, this will become a B positive. Got my point? This test was very simple, right? We need to take a known antibodies. Now my question was, it's not about ABO grouping. How the hell in the first place we created the known antibodies? How we created known antibodies in the first place to utilize them as a reagents for my test. Antibodies are something that is made in the humans or in a living organism. How we synthesized them? Is it possible for us to synthesize an antibody?
Yes, sir.
How? How then? Do you tell me that?
In human body it's impossible.
It's simple.
In human body it's impossible, but in animals.
In human body it's impossible. It's possible. That's why in COVID patients, recovered COVID patient serum we given to the patient. Right? Everywhere it's possible. Listen, very good. Anyway, see. Now I said listen carefully. Now, very simple. Now what I will do was, for example, I will take a known patient. And by the way, there is a Karl-Landsteiner law. The law states that if a particular agglutinogene present in the patient, the corresponding agglutinin is absent in the plasma. If you still can't understand, simple. If patient has A antigen, plasma will have B antibodies. If he is B antigen, A antibodies will present. If he has both A and B, nothing will present. If he don't have anything, both A antibody and B antibody present. The opposite antibody is present. Clear? Now, let us assume he is a A group. Okay? I have taken his blood in a test tube and I centrifuged it. I got a serum. In serum, what antibodies are present from him? I said he is a A group individual. I took his serum, serum. This serum will have B antibodies. Am I correct? From the A group patient, if I take his serum, I will have B antibodies. Am I correct? Now I can use these antibodies to do the test. I can use this reagent. Am I correct? Now there is a challenge. The challenge was not only B antibodies but also all other nonsense antibodies will be there from all of us, like anti-hepatitis antibody, anti-TB, all antibodies are present in this supernatant, not only B antibodies. Sometimes they will react with some other antigens and they will give false positive reactions. I don't want such antibodies, right? Such antibodies are called as polyclonal antibodies. Polyclonal antibodies are not good. A mixture of many types of antibodies. They are not so specific. Got my point? Rather than I need only anti-B antibodies. How to obtain only one antibody? That's very tough. Such antibodies are called as monoclonal antibodies. Only a single type of antibody. How to get this only single type of antibody? How to get a monoclonal antibody? Got my point? So I given a very basic understanding about what is monoclonal antibody. Now I will tell you somewhat depth. Okay, this is one of your topic. That's what I'm teaching. Now listen, very very interesting. I have taken what is your favorite animal? Cat. Okay. This is a cat. Okay, your favorite animal, cat. Okay. Sorry if it is scary. Anyway. Okay, I have taken a cat. Okay. Now understand very clearly. This is our Gyan, right? Your name? Gyan. Yeah. Now this is our Gyan. Gyan was a group individual. And I know that Gyan will have a B antibodies. Okay. I have taken these antibodies in a serum. Now I will inject his serum to a cat. Okay. Will cat become immunized now? What happens within the cat? Will it get a super boost, super immunity to the cat? Will it become more antibody, powerful antibody cat? Now listen, this is a human. This is a different species. Even the antibodies in the human, these antibodies will become an antigen to the cat. Even though it's an antibody, this antibody will become an antigen to the cat's immune system. It will treat this antibody as an antigen. It's a foreign to the cat now. Got my point? So why I need to tell this analogy? Because simply just because of antibody in the human doesn't mean it's an antibody to the cat. It will become antigen to the cat because it's a foreign molecule to the cat's immune system. Now what cat immune system will do? It will treat it as a foreign. Now its immune system will get activated. Now it will have its own cat antibodies, B cells. Now cat B cells will make plasma cat B cells and they will make an antibody. These antibodies we are taking in the reagent, that anti-sera B reagent. These antibodies are called as monoclonal antibodies. And these antibodies we are going to do the test. It's not a direct step. We are from other foreign species. It can be a cat, it can be a monkey, it can be a rabbit, whatever. Not birds, okay? Parrots, or mammals we should take. Okay, simply. Clear? So they are called as monoclonal antibodies. So what is the benefit of having monoclonal antibodies? Monoclonal antibodies have a very, very high specificity. They are very specific to their antigen. But there is a problem with monoclonal antibody. They have a good specificity. There are two terms that we need to understand. The binding terms in the, if this is an antigen, this is an antibody. There are two important things. Affinity and avidity. Do you know what is affinity? And what is avidity? Even I forgot, I need to check this. Let me, affinity, is it AFFI? Let me check it. Affinity, somewhere I got. Let me take the help of affinity versus avidity. 1 second. Oh, I'm not getting this internet. Someone, just Google affinity and avidity and tell me the answer. What is affinity and what is avidity? Tell me.
Binding strength.
That is affinity. What is?
Both measures of binding strength.
Both.
Number of binding interactions.
You tell me, definition of affinity in immunology, in antibody. Tell me. Then I can teach you. Because I don't want to give you wrong answers.
So affinity refers to the strength of a single interaction between a single antibody binding site and a single antigen binding site.
Single antigen antibody is called affinity. What is avidity means?
It represents the overall strength of the antigen antibody interaction when multiple binding sites are involved.
Very good. Now I got it. Listen. See, affinity means the specific nature of the antibody to only attach to the specific antigen. Okay, this is called affinity. But since it only attaches, doesn't mean it will bind tightly to the antigen. It can easily, detach easily. So affinity might be high, but avidity is less. The strength of the binding is avidity. The specific nature of connection, attaching is called affinity. But the strength of this binding is called as avidity. The strength of the binding can be increased by using multiple antibodies of the same type. That is a major problem with monoclonal antibodies. Monoclonal antibodies have good affinity, but they have less avidity. Whereas polyclonal antibodies have less affinity but good avidity. Polyclonal antibodies will tightly bind with many antibodies will bind to the avidity is there, but they are not specific. Got my point? That is the difference between affinity and avidity. You should know this concept when we are studying the monoclonal and polyclonal. That depends on the from the application to application. Sometimes we don't need a specificity, rather than we just need to bind to the molecule. At that time we can go with the polyclonal. When we need a specific nature, then we will go with the monoclonal. For example, blood grouping, we must have affinity rather than avidity. Okay, yeah, that is the concept of monoclonal antibodies. That is clear to you. Then I just want to discuss one famous confusion among students. The famous problem in the beginning. I said one interesting thing, that is whenever I got to attend. See, whenever this is a blood, a pathogen released pathogen associated molecular substances, and they attract the macrophage destroyer. And also our immune system has some proteins, circulating proteins. These proteins are called as complementary proteins. Complementary proteins. They also play a vital role, very vital role, very important. I will tell you later what is that. Even I need to study this one before I'm teaching you. But anyway, some of these proteins are almost 13 or 19 types. C3, C1, C2, C3. I think 13. I think we have C1 to C13. There are 13 types of protein molecules. Together these protein molecules are called as complementary system. Complementary system. Among them, C3b molecules will bind to the pathogen immediately. The C3b molecules, they will also act as a red flag molecules to send a red signal to the macrophage. What exactly happened was I got a terrorist, but I'm a civilian. I'm not a direct police officer, so I have a spray paint. Immediately on the terrorist, I draw the spray paint. That spray paint can be a C3p molecule. That spray molecules will facilitate phagocytosis. This molecule mediated phagocytosis, C3b molecule mediated phagocytosis is called as opsonization. Opsonization is one of the important way of phagocyting the pathogen. Have you heard about opsonization? Yeah. Then the molecules that facilitated the opsonization are called as opsocins. Opsocins. The molecules that facilitated the opsonization are called as opsocins. And the process is, the process similar to phagocytosis is called as opsonization. Now I want to tell you one interesting fact. Okay, the confusion among you, misunderstanding, not confusion. Simply we thought this is a bacilli, E. coli, for example. Okay, E. coli has an antigen, an antibody binded, IgG antibody, and this antibody somehow miraculously killed the, killed the E. coli. It's not as easy as that. Now I want to tell you one interesting fact, a groundbreaking fact. Antibodies are useless. They can't kill the pathogen. We always thought antibodies will kill the pathogen. Antibodies kills, antibody kills. They won't. Antibodies are useless molecules. Got my point? Well, Vamsi sir, what are you trying to say? You might get, yeah. Antibodies, yeah, I understood your expression. Now listen, let me tell you. Imagine we have a gun. Is it gun? Oh, okay. This is a, it should be like, now it came. Yeah. This is a gun. Okay. I see. Now, this gun have bullets. Okay. Somehow I need to connect it. Wait. Yeah. Now, this gun will be holded by some person, right? Like this. This person who is, aiming, that aiming is given by the antibody. It will only aim the gun towards the pathogen. But actual killer is this bullet. This bullet will kill the pathogen. Okay. That bullet was complementary proteins. It is the complementary molecules that will kill the pathogen, not the antibodies. What is the use of antibodies? They will just guide the complementary proteins. They will just antibodies are acting like a guiders to a specific target. And the actual bullets are always complementary system. Got it? Actual bullets are complementary molecules. How they will do this one? You don't know this fact. There is a series of C6, C7, C3, C2, A, C3B. There is a system of a complex system will be formed by the complementary system. Once this system formed, what they will do? They will start polymerizing. They will start drilling holes in the pathogen surface. To the lipid cell wall, they will start drilling holes. Now these drilled holes, what happens? Once the holes are drilled, the inside materials will come outside and it will die. This drilling hole is one of the most potent, lethal, the worst way of killing pathogen. It's the nightmare to the pathogen. And this mechanism is called as membrane attack complex. Very, very dangerous. MAC will kill the pathogen. And that MAC will be triggered by an antibody. Antibody will trigger the MAC and MAC will kill the pathogen. MAC will kill the pathogen. Okay. MAC will kill the pathogen. So MAC kills pathogen via antibody guidance. That means this was a very famous experiment done by Paul Ehrlich, very greatest scientist. Paul Ehrlich, something, Paul Ehrlich. He got Nobel Prize. Okay, one of the biggest prize in the medicine to a scientist. It's like Shah Rukh Khan level celebrity in science, right? Yeah. So Paul Ehrlich or PMOD, if you guys. Paul Ehrlich, what he did was this Robert Koch, Edward Jenner, vaccines. They are saying there are antibodies they are doing. What he did was he took the anti-serum. Okay, he took a petri dish. There are some pathogens. Then when he added antibodies, the pathogens died. Clear. Waste pasture was clear. Vaccines are working. Then what he did was he heated the sample around 37 to 40 degrees centigrade. Then after heating this antibodies, he again tested the antibodies. Then somehow antibodies didn't kill the pathogen. Pathogens are still alive. They are very happy. Why? Why the same antibody? Then he immediately went for checking the antibodies, and antibodies are present. Antibodies didn't denature. But why? After slightly heating the serum, why my antibodies didn't work? So he immediately said there is something else, heat sensitive proteins, heat labile proteins that are helping the antibodies to facilitate the pathogen killing. There is something complementary to the actual immune system, and that complementary things are helping. Definitely those complementary systems have a very small proteins, heat sensitive proteins. Once they started researching, they came to know there is a very big game called complementary system game, and that is happening inside the body. And very interestingly, out of 100% human genome, 70% of human genome dedicated to regulate the complementary system. They were shocked. Not even for making brain or heart. 70% of human genome completely dedicated to maintain the complementary system regulation. So complementary system is like a grenade, it's like a nuclear bomb which is always ready to explode, the MAC complex. See, as I said in the beginning, even before, just when the pathogen entered in the system, the C3b molecules attached for opsonization. Not even antibodies that came into the story. It is the C3b, that is the complementary proteins came into the action first. They are always ready, irrespective of anyone. Pathogen and outside something came inside, game over. If they will try to trigger, they will try to attack. So always, or simply we can say, our antibodies are always regulating complementary system. Antibodies' role is only to regulate complementary system. Please don't explode. Antibodies are keep on suppressing complementary system. If antibody is not suppressing, they will do whatever chaos they want in the body. Now we are seeing a new perspective of immune system. See, till now we thought only antibodies. Actually, 70% of genome dedicated to suppress the antibody, to guide something else. The more dangerous guy is there in the body, that is complementary system. I said previously, wear and tear happened. Whenever the wear and tear happened in the synovial joint, it's not the wandering macrophage that came. Actually, the C3b molecules, they binded there. Oh, you are not a good guy. They binded automatically. Okay, I started my YouTube channel just to, even I'm not getting sufficient time to teach complementary system in the classrooms. So I went back to my room and I took almost 2.5 months to understand complementary system in my masters actually. In masters I came to know complementary system. And I came to know that complementary system plays a vital role in TB because there is TB Research Institute and macrophages, antibodies. and I was trying to work on vitamin D activity. Vitamin D again a biochemistry. Then I was trying to understand vitamin D activity on macrophage, how this macrophage attacking the TB via some cytokines it's releasing through complementary system. Then I got another challenge. C3, this vitamin D metabolization happening by some enzymes, vitamin D enzymes, and there is one particular enzyme called CYP27B1. Now I need to understand that enzyme, something that is biochemistry connected, but working on a macrophage in the system of immunity against a microbiology. I thought, okay, I can understand, I can handle this complicated story. Then I entered somehow, into the domain of molecular biology. Now I need to understand the genes of this CYP27B1, the gene that is coding the enzyme. Oh my god, then I came to know the gene has 200 million base pairs in the internet bank, SNP banks. And there are around 11 SNPs, single nucleotide polymorphs or alleles in the gene pool. And I don't know what allele present in Indians. And I have 11. Out of this 11, I need to take only one golden thing to support my research. So blindly I tried with some literature, some people saying some connections. So I ruled out with one of the restriction site, that is RS118204012, which is completely a research molecule. No one did this, that's what research, right? So I asked my institute to provide the particular primer to attach to this particular gene, and that primer will cost around 4.5 lakhs. Since it's a government institute, they brought it free of cost to me, because out of 90 applications from my Loyola, only one person got approved to work in ICMR, and that is myself. It's not good or bad, I don't know, because now I'm tackling very challenging thing. And another challenge I got when I'm doing my PCR, I don't know the melting temperature, the annealing temperature of this particular primer, because it's none of the literature discussed about this one. So it's taking 1.5 months for me to get my PCR product, and the people I was misusing the reagent because I'm not getting result, and they brought for 4 lakhs. So they started doubting my capabilities because they thought that I was doing some mistakes in my master mix preparation or somewhere. Yeah, it's painful. Then my guide also started, Vamsi, can you or do you want to shift the domain? Yeah, it's like everyday pain. Morning 7 o'clock I'm going, my product will get around evening 7:30. 12.5 hours I need to do multiple steps in the lab, and I'm not getting result. Oh my god, that's too much painful. And at one fine day I got my PCR, bands in my, I came to know my, melting temperature. That's how science works. Something my interest on complementary system landed me onto complementary system mediated vitamin D regulation on macrophage for tuberculosis. Yeah.
So have you done your final year dissertation maximum?
I did in ICMR NIRT, and my paper got published. There is only one journal taking care of the genes, okay? And the journal was the gene, and my paper was published recently. It took 4.5 years to get published. And I got a very good results from my RS118. Luckily, I selected the right one.
It was your final year dissertation?
Yeah, final year dissertation. Yeah, I finished it. Yeah, I carried it to my PhD, but PhD I'm teaching and I'm unable to do my lab work. Yeah, it became challenging. I'm not doing my lab work till today because I need TV lab. It's challenging, and I have a 3-year villain in my home. I mean my son. No, with that kid, I can't leave him. Yes. All right. Okay. We'll see. So that's how you should be always eager to study, not only for today or tomorrow exam. You should connect these things and you should go for research. What you can do later. What is the gap in the research? So you should have that curiosity always in your minds. Not simply criticizing, no one will teach us. No, what are you doing yourself, by the way? That's important, right? If you ask, I love Michael Jackson, the singer, because there is one song by Michael Jackson called Man in the Mirror. Such a beautiful song. If you want to change the world, take a look at yourself, then change the man in the mirror. If you change the man in the mirror, the world will become a better place. To change the world, first we need to change ourselves. First we need to take the right action. We cannot blame others. You do. What we are doing? And you know what I did to my son? His name is Michael. I'm a Hindu. Okay, yeah. I asked my mom, I given one opportunity to my mom. Blindly, I don't want this astrology, nothing. I asked my mom and she said Surya Narayana, her father's name. So I took that Surya from her suggestion and my Michael, his name is Surya Michael. Yeah. Yes, passion matters in every aspect of your life. See, all this, an MLTN, now I'm in a central government permanent job, next 30 years I will be here. That because of passion on our subject. I didn't just, my ma'am torturing with this complementary system, what to study. If I thought like that, I might not be here today to tell the same story to you. So you are in that stage. I did complementary system when I was there in my second semester of MSc Masters. So you are on that same golden time. Utilize it properly. Try to go to any nearby government institutes. No one will refuse an intern. Show your capability. See their what work they are doing. Similar similar research what you draft. Request them, definitely they will help you because they need a manpower. At the end of the day, we all need labor or someone to help our work. Since you already understood their work, they will be very happy. You say, I don't want a single penny. Then they will be, a free labor. They will allow us. But we will get the benefit of collaborating with them. Then we will request them politely. Can I do a small work, to complete my masters? No one will refuse when we are getting a degree. Right? So you need to impress the persons. So identify your area, what you are interested in, microbiology, bacteriology, virology, everything is there. Just identify your passion, work on it, and you can, this is the right time. Your decisions today matters. So thank you very much for today. Thanks so much.
Thank you.